Hello everyone, Mark here. I thought that before heading into the lab to do today's experiment, I would give a little bit of background theory into the alcoholic fermentation process in yeast, since that's what we're going to be doing in the lab. So you may or may not know that yeast belong to the domain eukarya, so the eukaryotic cells, and they are unicellular, so single-celled organisms, and they belong to the kingdom of fungi. These yeast, the particular one that we're using today, are commercially very important in that they allow us to make wonderful things such as bread and beer. So the yeast species that we're going to be using today is called Saccharomyces cerevisiae. And its more common name, and you'll see it if you go to a store into the baking aisle, it's called baker's yeast. It can also be used to make beer using the fermentation process, and in fact its second name is cerevisiae. Cerveza is the Spanish word for beer, so that's where that came from. So what we're looking at here is alcoholic fermentation, and this is an anaerobic process. So it's in the absence of oxygen. And basically there's two major components to this. So the first one is glycolysis. It's basically the conversion of glucose into 2 pyruvate molecules. So for every 1 glucose molecule, we'll yield 2 pyruvates. And this is actually a 10-step process, and it involves 10 separate enzymes that convert glucose bit by bit from a 6-carbon molecule into 2 times 3-carbon pyruvates. So that's the first step, so that's glycolysis. The breaking down, the lysis of glucose, glycolysis. The second step is the fermentation step. And what this is going to do is going to convert these 2 pyruvates into ethanol, so 2 ethanols, and carbon dioxide. And this ethanol is a 2-carbon molecule. So, and this step here involves 2 steps, 2 enzymes, and basically yields carbon dioxide and ethanol. So it's the carbon dioxide that helps the bread rise, and the ethanol created is what you would use for beer. More on the carbon dioxide later. So why is this process necessary, you may be asking? Well, the reason for it is so that yeast can produce ATP in the absence of oxygen. So this is an anaerobic process. I.e. done in the absence of oxygen, and what we're trying to produce here is some cellular energy, some ATP to help drive cellular processes. Now, normally, in the presence of oxygen, what happens is the pyruvate that is produced from glycolysis will be moved or shuttled off to mitochondria to participate in the Krebs cycle. You might have also heard this called the citric acid cycle. So this is occurring in mitochondria. And this requires oxygen. The other product of glycolysis is a substance known as NADH, and we're going to get into that in a little bit. And what NADH does is it gets shuttled off to the mitochondria, and it's able to participate in something called the electron transport chain. And together, these two mechanisms in mitochondria are capable of creating 36 ATPs per glucose. So for every glucose that entered glycolysis, in the end it's going to yield 36 ATP as a result of the Krebs cycle and the electron transport chain. Most of it's going to happen from the electron transport chain. However, we're talking about an anaerobic process, so this doesn't happen. So instead, we have glycolysis plus fermentation. And what that will yield is 2 ATPs. Per glucose. The actual process of glycolysis requires 2 ATPs to kickstart the first of the series of enzymatic steps, but then after that it creates 4 molecules of ATP, so you end up with a net gain. So it's a net gain of 2 ATPs per glucose going into the glycolysis reaction. Now you may be wondering, well, if glycolysis is making ATP, then what's the point of the fermentation step? Well, the reason for that is due to this molecule here. We have NADH, and NADH is a species of a cofactor known as nicotinamide adenine dinucleotide, or NAD. During the process of glycolysis, NAD plus is reduced into NADH, and that's the NADH here that in the presence of oxygen would go to the electron transport chain, but we're talking about the absence of oxygen here. So this is a reduction reaction, and so in order for glycolysis to start over, we need to replete or put back some of that NAD plus. So what happens during the fermentation step is that the fermentation step allows the oxidation reaction to occur. From NADH back into NAD plus, thus replenishing the NAD plus to drive another round of glycolysis. That's going to create more NADH, and then that fermentation process will then oxidize that back to NAD plus so that another round of glycolysis can occur. And this can go around and around in circles and generate 2 ATP for every circuit it makes around this process. So let's quickly just review that in a little bit more detail here. You can see here's a schematic of the whole process, starting with glucose in glycolysis. And remember that this dotted line to pyruvate is a 10-step involving 10 enzymes. And basically what happens is, in the remodeling of glucose and the splitting up of glucose, two hydrogens get removed from glucose and they get transferred to the NAD+. So what happens if those two hydrogens are basically made of 2 electrons and 2 protons, remember your chemistry class, and of those the 2 electrons and 1 of the protons will get accepted by the NAD+ as part of the enzymatic conversion. So what you end up with is NAD+ plus 2 electrons plus 1 proton, and that's going to give NADH, the reduced form. The other proton is actually sent off elsewhere in the cytoplasm to do other roles, other parts of other metabolisms. The only issue here is that if you get to this stage, you need to replenish this NAD+, simply because you won't be able to carry out glycolysis again without it, because it has to be there to accept those electrons and 1 proton. So the way that it's done is here in this 2-step process. We have a electron acceptor known as 2-acetaldehyde, and what it'll do is it'll take back those 2 electrons, and thus our NADH will be oxidized back into NAD+, and therefore we're back to the start and another round of glycolysis can occur. The end product of this is ethanol in a yeast cell, and also carbon dioxide is given off, and that's what we're going to be measuring as a guide to how well this process is progressing in our yeast in the lab. You may have heard of this mechanism in humans. Instead of making ethanol, we make lactic acid in a 1-step process. So in humans, lactic acid is produced. And it's for the same reason, to replenish this NAD+ by oxidizing it and having an acceptor molecule of the 2 electrons. You'll know about this if you play soccer or you sprint or you play any sport with high quick bursts of activity, that sometimes the demand for oxygen to your muscles is so great that they're not able to get enough oxygen to go through the normal processes of the electron transport chain and Krebs cycle to get ATP, and instead they'll have to go the anaerobic way. So as you know, you're not able to sustain this very long simply because of the byproduct lactic acid. It causes this burning sensation in your muscles, and too much of that can actually lead to acidosis as well. So and we need to use oxygen as an oxygen debt to remove that lactic acid from our system. So basically that's how humans deal with anaerobic respiration. In yeast, they're able to make alcohol, and as I said, this is a commercially kind of good thing for us, because then we can make wine and beer from this reaction, and also use the carbon dioxide here for helping bread rise during the baking process. So what does that mean with respect to our experiment? Well, simply put, what we're going to do is we're going to examine this whole pathway by taking some glucose And we're going to add that to some active yeast that we've bought from the store, baker's yeast, and we're going to measure the amount of carbon dioxide produced. And the reason for that is because when the carbon dioxide is produced, that shows us that the fermentation process has been completed. So it's directly proportional to how many rounds of glycolysis that has been able to proceed as a result of putting glucose with the yeast. So what we'll have are a bunch of different tubes. One of them is going to contain glucose and yeast, and we'll measure how much carbon dioxide is produced. Another one is going to contain glucose, yeast plus magnesium sulfate, and this is because magnesium has been shown to be helpful with some of the enzymes of glycolysis. So we'll see whether adding magnesium sulfate changes the amount of carbon dioxide produced. We'll also be adding an inhibitor of glycolysis. So sodium fluoride is an inhibitor of glycolysis. And we'll use two different concentrations of that inhibitor to see what happens, whether increasing the concentration changes the amount of carbon dioxide produced or not. And another tube will have pyruvate and sodium fluoride in it. So I'm not going to tell you what that one's all about. You'll have to kind of think that one out for yourself. So it's got pyruvate, sodium fluoride, glucose and yeast in it, and we'll be looking at how much carbon dioxide is produced. So be thinking about what the purpose of that tube is. And the final tube will be actually a negative control with just water. So without further ado, I'll let you see me in the lab doing the experiment, and what's going to happen is I'm going to be sending you some raw data so that you can average that to create a nice graph that'll help you explain the results and hypothesize and understand this whole process.
Hello and welcome back to the biology lab. This is now lab number 3 and it's the alcoholic fermentation of yeast. So here's our yeast. It's actually in a suspension, a 5% suspension, and basically that's just warm water with store-bought dry active yeast that's used in bread making. So what we're examining are the steps in alcoholic fermentation, and you'll have seen in theory that that begins with glycolysis. So it's the breakdown of glucose eventually into pyruvate, and that yields ATP for cellular energy. After that, pyruvate is then converted into alcohol and carbon dioxide. And it's the carbon dioxide that we're going to be measuring today in these fermentation tubes. So basically, as you can see, I've got a bunch of different solutions here to set up 6 different tubes. All the tubes are going to have 5% glucose in them, since I mentioned that the process starts with glucose, breakdown of glucose and glycolysis. In some of the other tubes, we're going to have a cofactor known as magnesium sulfate. And magnesium sulfate is a necessary cofactor that helps some of the enzymes in the process of glycolysis. We have an inhibitor called sodium fluoride. This inhibits glycolysis. And we also have pyruvate that I just mentioned that gets converted into alcohol and carbon dioxide. So in your handout, you'll see the ingredients for each of the 6 experimental tubes. So just to go through them very quickly, I won't bore you by filling them all in front of you, but just to go through them, we have tube number 1 that's going to have 2.5 ml of glucose, 5 ml of water, and 10 ml of the yeast. Next up is tube number 2. This one's going to have 5 ml of magnesium sulfate, 2.5 ml of the glucose, and 10 ml of yeast. Tube number 3 is going to have an inhibitor. It's going to have 0.5 ml of the sodium fluoride, 2.5 ml of glucose, and 4.5 ml of water, as well as 10 ml of the yeast. Tube number 4 is going to be 10 times more of the inhibitor, so we'll see whether there's an effect there. We're going to have 5 ml of the sodium fluoride this time, with 2.5 ml of glucose and 10 ml of yeast. Tube number 5 is the pyruvate tube, so this one contains 2.5 ml of pyruvate. It also contains 2.5 ml of the inhibitor, sodium fluoride, and 2.5 of glucose, and 10 ml, of course, of yeast. And the final tube will contain 2.5 ml of glucose and 15 ml of water, so no yeast in tube number 6. So in a second I'm going to show you how the tubes are filled, and then we'll fast forward through all the filling so you don't have to sit through it, it's a bit tedious, and then I'll show you the experiment and measurements. Okay, so let's just show you how to fill one of these. So I'm just going to do tube number 1 for now. This one's going to contain 2.5 ml of this glucose. We'll use these pipettes. Right. And this one also has 5 ml of water, so obviously I'm going to use a fresh pipette. Water. And finally 10 ml of the yeast here. Simply add it to the flask like that. So before we start the experiment, I'll invert the flask to get the liquid up into the top, but I won't do that until I've filled all of the tubes. So there's one, and I just have to go and fill the rest of them. So I think what we'll do is we'll fast forward through this step, and we'll see in a second with the 6 tubes ready to go for the experiment. Okay, so the tubes are filled with all the different ingredients. Now it's time to get the mixture up into the tail of the tube, and you simply just tip it like this. You see that it starts to form bubbles. Eventually, all goes up into the top of the tube like that. So I'm going to do that for each of them, and then I'm going to make a little mark where the bubbles started, and then we're going to get them into the water bath. Right, so our tubes are now in the water bath, 37 degrees C, and that's going to kick-start the process of glycolysis. So the experimental tubes that are able to create carbon dioxide, that's going to be represented by a bubble forming in the top of the tail of the fermentation tube. So I'm simply going to be measuring every 5 minutes the amount of carbon dioxide produced, and that will give us an idea about how well glycolysis and the alcoholic fermentation of yeast has progressed. Okay, so the experiment's over. I'm just going to quickly show you the tube. So you can see quite a big air bubble in this one at the end of play, from here to the marker made up there. It's tube number 2. See, even bigger one here. Goes from here to the marker made up about there. 3. This one from here to the marker I made around about here. I made all the measurements after every 5 minutes. Not so big here. This one from here until the mark I made just there. Big one in number 5. From here to the bottom all the way to the top, because it was full to the top, this one. And of course number 6, there was no change. So what I'm going to do is I'm going to do the experiment again without the camera, and I'll be more careful and less sloppy. Obviously the way I kind of filled them, I should have added the yeast last so that nothing kind of started before we got going, and add each of the ingredients if repeated to the different tubes. So I'm going to do that. I'm going to make a number of new recordings, and we're going to be sending you the data so that you can graph it and start to answer questions about this process. Oh, sorry I didn't see you there. Anyway, so that's the end of lab 3 on the alcoholic fermentation of yeast. Now you can go to Moodle, where you'll be able to access the raw data that I collected during the experiment, and you'll be able to create a graph so that you can submit that to the Moodle quiz, and also answer questions all about the experimental procedure and some of the data. If you have any questions about anything to do with the lab or biology in general, you can always send myos to your teacher, to Mackenzie, to Boris, or to me, or you could join any of us during our office hours. And finally, I am the TLC representative for biology, so check your course outline, you'll see the schedule on there, and you can join me on my Zoom channel if you want to talk about anything to do with biology, the course, the lab, or whatever, so don't hesitate to join me on that. Okay, so get off to Moodle and get on with your questions, and hopefully we'll have another one of these for you next week. So until then, take care, be safe, I'll see you. Cheers.